All right, so it should look like I'm about to edit it. And then now it should be full screen. Is that right?
That is not correct.
Okay. Let's see here. Try it a different way.
You could, if you want, make just the main slide. If you're having trouble, you could just make the main slide bigger and then it'll help a little bit.
Let's try this. How about now?
You're good now.
It's full screen.
Full screen.
It moves. Okay. All right.
Okay. Great. Okay, so I'll introduce Ali. Hi, everyone, again. Today, we're hosting a great colleague and a friend, Ali Cohen from University of Pittsburgh. She's an expert in lipoprotein metabolism and the interaction between chylomicrons, chylomicron metabolism and inflammation. And she will tell us about Tregs and triglycerides today. Ali, it's all yours. Thank you.
Thanks, Lale. And it's so nice to see everybody. I know this time of year would normally be me looking forward to ATVB and, you know, all the social piece. So it's good to see you guys. And I'm looking forward to seeing everybody in person again. I want to throw out that, like, whatever our next meeting is that's in person, I think we should all agree to come a day early and stay a day late and, like, just do something really fun, you know, just we can camp out in the presidential suite a la that ATVB years ago, right, Katie? So, all right, so I'm really excited to present today. For those of you from Ottawa, you heard me give this presentation a week or so ago. I hope second time around is also good. I want to normalize not having brand new presentations for every single seminar that you give, in part because that makes me feel a little bit better, but also because really this work is really the state of the art for our lab. It's really where we're at, and it's the kind of things that we're thinking about. We are really interested in, as Lale said, we're really interested in lipoproteins and lipoprotein lipids. And today I'm going to talk about Tregs and triglycerides and whether a finding that we have in the gut is also good for the heart. As a physiologist, I'm often really thinking about both a narrow physiological niche, but then big picture, how do changes in one anatomical location broadcast to the rest of the body? Because ultimately, the things that happen in one place tend to happen elsewhere. I think the best examples of this are in the current cancer immunotherapies, where we seem to have a really effective mechanism for curing certain forms of cancer by ramping up the immune system. But then big picture, you also ramp up the immune system in the heart. And a lot of work has shown that you end up with myocarditis, atherosclerosis, and whatnot. So you've got, you have this crosstalk between different anatomical locations. We think that we have a mechanism for lipid sensing in gut Tregs, and that may also play out in other locations. I'll start with our general point of view, which is starting with triglyceride-rich lipoproteins. So TRLs, triglyceride-rich lipoproteins, are chylomicrons and VLDL. They come from the liver and they come from the intestine. And these are the physiological lipid fuel. These contain triglyceride and cholesterol in their core. They're bounded by phospholipids. And then they're decorated with apolipoproteins, which are shown here. ApoB is the structural apolipoprotein. We're very interested in ApoC3, and I'm going to talk about that a bit later. But what you can see is that these triglyceride-rich lipoproteins have a very specific size and structure. And if you compare that to purified free fatty acids or fatty acids that are released from lipid stores like adipose tissue or intramuscular fatty acids, these are found in the blood bound to albumin. And ultimately, when they enter the cell, it's through a lipid receptor. But I always try to point out, you know, when we're talking about fatty acids, this is what we're talking about. We're talking about, you know, a palmitate, a single palmitate or a single oleic acid, something like that. When we're talking about triglyceride-rich lipoproteins, we're talking about a core of complex lipids that need to be metabolized and a protein structure on the outside. They're very different vehicles. Functionally, triglyceride-rich lipoproteins carry at least 10 times the amount of fatty acid, fatty acyl groups as the amount of fatty acids that are released from adipose, even when you compare the fasting state when these are low and when these are high. So, you know, this is where your body gets its lipids from primarily. So as I said, triglyceride-rich lipoproteins are only made in two locations. The intestine, the small intestine, makes a chylomicron particle. The liver makes a VLDL. Now, these lipoproteins deliver lipids throughout the body, and they are ultimately cleared by the liver through primarily the low-density lipoprotein receptor, LDLR, though any of you who are familiar with lipid metabolism know that there are many other lipid receptors that play really important roles in taking up remnant lipoproteins and metabolizing them and clearing them from plasma. Now, our favorite lipoprotein, apolipoprotein, which is ApoC3, is a critical regulator of all of these processes. And that's why we first became interested in understanding the physiological role of ApoC3. So ApoC3 was first discovered, you know, over 50 years ago as an inhibitor of lipoprotein lipase on endothelial cells. Lipoprotein lipase can cleave fatty acids from the core of a chylomicron or a VLDL, and then those fatty acids get taken up into the cell. So ApoC3 can inhibit that action. ApoC3 also inhibits low-density lipoprotein receptor, and in doing so causes remnant lipoproteins to accumulate in plasma. And so as a result, high plasma ApoC3 levels are directly related to high plasma lipid levels. Intracellular studies have shown that ApoC3 also increases the synthesis and secretion of VLDL from liver, which again would increase the amount of plasma triglyceride and cholesterol present during high ApoC3 states. And so when I started this work, we were really interested in following up in the small intestine and asking what ApoC3 does there. And we were really surprised to learn that actually high levels of ApoC3 in the small intestine decrease the secretion of chylomicrons into lymph. And we spent a lot of time, pretty much the last 5 years, really describing this and finding that ApoC3 is working on an enterocyte-specific intracellular metabolism to cause those cells to actually utilize and burn dietary fat rather than package that dietary fat into chylomicrons. And so this translates to a decrease in chylomicron burden after a meal. But what you can see is that ApoC3 is at all the branch points of plasma triglyceride metabolism. Because of this, ApoC3 plays an important role in disease. So ApoC3 causes hyperlipidemia in humans by inhibiting triglyceride clearance and extending the postprandial period. Again, that's because of its inhibition of lipoprotein lipase and Ldlr. We know in humans that plasma ApoC3 levels are an independent risk factor for cardiovascular morbidities and mortalities. Again, if we are walking around, the dogma is that we would like to have less ApoC3. That puts us at a decreased risk of cardiovascular events. Frank Sachs' group did a nice job showing that HDL containing ApoC3 are actually associated with an increased risk of cardiovascular disease rather than cardioprotection, which really is in line with what we understand about HDL, that it's not a single entity. Increasing plasma HDL or HDL cholesterol is not a simple, magic bullet to curing cardiovascular disease, but that instead there are protective pools of HDL and there are non-protective pools. And ApoC3 seems to be one marker of a pool of HDL that doesn't actually help. And so, together, because of these reasons, ApoC3 is a drug target for reducing plasma lipids and cardiovascular disease. And one of the major ways that ApoC3 has been targeted is through an antisense oligonucleotide through the Ionis pharmaceutical company. Their ASO was volanesorsen. Again, they were really proving the principle that lowering ApoC3 in plasma would be a great adjunct therapy for people who are already on statins but still have high plasma lipids. And so therefore they could reduce their cardiovascular disease risk. That has really since evolved into, instead of using an antisense approach, using an antibody approach to ApoC3 in plasma, again, to lower triglyceride-rich lipoprotein levels. And again, that's through statin pharmaceuticals. And I think this is pretty much where we're at right now with ApoC3 drug targets. The role of ApoC3 and lipoproteins, however, really extends beyond simply total plasma cholesterol and total plasma triglycerides. And I first became really interested in that extension because of the CANTOS trial and really understanding that cardiovascular disease is a disease of both hyperlipidemia and inflammation. So we know that, and certainly this group is well versed in this, we know that the people who drop dead of a cardiovascular event are more than likely to already have well-controlled plasma cholesterol levels through statin therapy. We already know that lowering plasma triglycerides and cholesterol is protective, and yet there's a large subset of people walking around dying of cardiovascular disease who seem to have residual risk factors. And the CANTOS trial was the first to give us, a large-scale idea that lowering pro-inflammatory cytokines could prove the principle that of the chunk of that residual risk that was due to the immune system. And so this is just kind of a collection of findings, by no means the only ones, just that I was reading that were telling me, okay, we need to understand more about the immune component and where hyperlipidemia may impact immunity and how that may also play a role in cardiovascular disease. And so that led my lab to this hypothesis. And it was really mostly out of curiosity that we really went down this road. So we asked, do immune cells, and particularly immune cells that are important in cardiovascular disease, which for us was T cells and Tregs. Do these immune cells endocytose triglyceride-rich lipoproteins like a hepatocyte does? Do they take up that lipid the way that all other cells in the body are able to take up that lipid? And does ApoC3, through its metabolism of lipoproteins, regulate immune cells? And so again, here's our hypothetical model. Here's a generic immune cell. I'm going to talk a lot about Tregs and T cells, but the idea is, you know, the immunometabolism world has done a really nice job showing the relative contribution of glucose metabolism and fatty acid metabolism in immune cells, but whether triglyceride-rich lipoproteins with their abundance of both cholesterol and triglyceride can be utilized by these cells, whether this is a receptor-mediated event like it is in other tissues, and whether the apolipoproteins regulate this was really unknown. And that's what we set out to find, and I'm going to tell you about today. So for much of these studies, we've used the human ApoC3 transgenic mouse model. These were originally made by Jan Breslow's group. And you know, many of us know Jonathan Smith. He's on that paper. And we got these originally. They're now also at JAX. You can order these live. I will say that our colony is on a defined background. We actually backcrossed our colony onto the C57 Black 6J background. The animals at JAX are on a mixed background, which is important to know for immunological studies. We also swap dirty cage bedding between our experimental and control mice for 2 weeks prior to all of our experiments. And we do this to normalize the microbiota between groups. It's not perfect, but it at least lets us know that our control and experimental animals are at least exposed to the same microbial communities. And then we also use male and female litter mates. We haven't seen a sex-specific change in any of these findings, but we usually in the first set of experiments include both male and female so that we can capture that potential sex as a biological variable. And so a little bit more about our model. This is ApoC3 mRNA expression in the liver, where we know that ApoC3 is primarily made, and then from the proximal to distal length of the small intestine. And I'm showing in the hatched bars the endogenous mouse ApoC3, which is still present in our transgenic mice, and then the human ApoC3 here in gray. And so we get approximately 5-fold human ApoC3 expression in the liver and then along the gradient of the small intestine. And we know that these two genes, mouse and human, do not actually regulate each other. So they're both expressed. As we would expect, our ApoC3 transgenic mice have very high plasma triglycerides. They have so much ApoC3 coating all of the triglyceride-rich lipoproteins that they accumulate in plasma, and it takes a very long time for them to clear into the liver, again, because of that inhibition of LPL and Ldlr by ApoC3. So often after a meal, if we take blood, these animals have a pink Pepto-Bismol-like plasma. They also have high circulating total cholesterol. And I will come back to this after I tell you my Treg story, but you know, if I go back, look at these triglycerides, ultra high. Look at these cholesterols, high. And so one thing that I thought when we started working in the ApoC3 transgenic mice was, you know, I always wondered why the cardiovascular disease community didn't use these mice as a CVD model. I actually felt kind of like everybody uses Ldlr knockout, everybody uses the ApoE knockout. Why aren't they using, you know, our wonderful ApoC3 transgenic mice, especially because of how closely related in epidemiological studies ApoC3 is to cardiovascular disease. So when I first started my lab, I had as an idea, this pet idea that I would like, show everybody why this is such a great cardiovascular disease model. I'm going to now convince you why that's probably not the case, hopefully by the end of this seminar, but that's something I was definitely thinking about. So we asked first, just proof of principle, do immune cells have these receptors that are required to take up triglyceride-rich lipoproteins? And so we started with Tregs from the spleen. You'll have to excuse some of our lab's naivete with immunology. We really started and have gained a tremendous amount of expertise. So we thought spleen, that's where immune cells are. We'll just start there. That is definitely not, it's not as simple as that, but these are splenic Tregs, which we isolated and sorted. And we looked at some basic transporters, CD36, LDLR, LPL. They're there. There's no real difference in expression between our wild type and ApoC3 transgenic mice. Then we used an LDLR antibody to look at LDLR protein expression. And we found that actually in spleen, there's a very low number of LDLR positive Tregs. Same in skin draining lymph node. But as you get closer to the gut, you actually increase the proportion of the population that expresses LDLR. So by the time you get into the mesenteric lymph node, it's significantly higher. And then as you're into the intestinal lamina propria, there's a significant quantity of LDLR positive Tregs, which was really interesting to me because obviously the lamina propria is also the site of chylomicrons flooding into the lacteals after a meal. When we did the comparison of LDLR positive versus LDLR negative Tregs, you can see that in the spleen, there's a significant proportion of LDLR negative. And again, as you move towards the intestine, the majority of Tregs actually express LDLR. We've been noodling around with cell culture models, trying to understand the difference between naive cells that we isolate that are essentially ex vivo and induced naive cells and Tregs. And so what we find when we look for LDLR is that as you get into the induced Treg cells, we have an increase in both mRNA and protein. This tells us a little bit about the stability of LDLR expression and when it's highest and in which cells. We're still really actively trying to parse that out. This is just a post hoc analysis of a single cell RNA-seq data set taken from this paper. And my postdoc Jitendra went back and specifically looked at LDLR expression and found that in their analysis between natural and induced Tregs, there is a robust increase in LDLR expression. And so we just did a proof of principle experiment. We asked whether CD4 positive T cells, the parent population of Tregs, are capable of endocytosing triglyceride from triglyceride-rich lipoproteins. And so I'll take a second to explain this experiment. So what we did is we used donor mice to which we did a Bodipi triglyceride gavage. And then later we went and collected those Bodipi positive chylomicrons. So those chylomicrons become enriched in Bodipi. And then we took CD4 T cells from donor mice. And so we have a couple of different flavors here. We have wild type actually in this experiment, they're all wild type T cells, but one group of T cells received wild type chylomicrons. One group of T cells received chylomicrons from ApoC3 knockout mice, that's shown here. And one group of wild type T cells received knockout chylomicrons to which we added recombinant ApoC3. And the reason we did that is between the knockouts, between the ApoC3 transgenics and other mice, there's a change in chylomicron size, which can really impact chylomicron metabolism. So we wanted to make sure we were looking at normal sized chylomicrons with an equivalent amount of triglyceride. That triglyceride is labeled with BODIPI, and then asked whether the addition of ApoC3 would inhibit uptake. So what you can see is that compared to wild type BODIPI uptake in these T cells, knockout chylomicrons deliver more BODIPI labeled triglyceride, and the addition of ApoC3 inhibits that effect, which is exactly what we would expect. If these were hepatocytes, we would see the exact same pattern. So then we asked whether in vivo, this inhibition of triglyceride endocytosis would shift the metabolic program of Tregs. Presumably, they no longer have complete access to triglyceride from lipoproteins. And so do they adjust their intracellular metabolism? So these are splenic Tregs, which we isolated and then looked at genes of the lipolytic and beta oxidation pathways. And what you can see is in the ApoC3 transgenic mice, there's an increase in these genes compared to wild type mice, really suggesting that there's a shift in intracellular metabolism. Now, gene expression does not equal flux through these pathways. So we used seahorse metabolic flux analysis to ask whether the inhibition of triglyceride endocytosis would shift the metabolic program. And indeed, what we find is that early in the analysis, when we're looking at oxygen consumption rate, there is an increased capacity in our ApoC3 transgenic mice to for their basal respiration. Again, consistent with the idea that these cells, sorry, my Zoom just like popped up a funny thing. Consistent with the idea that these cells are responding to a difference in triglyceride substrates. And so we asked whether the change in both cellular respiration and triglyceride transport would actually have a physiological role. We looked at plasma cytokines to start, and I very naively thought that since ApoC3 transgenic mice have such high plasma lipids, and since ApoC3 is a drug target for cardiovascular disease, and because of the epidemiological studies that link C3 with human disease, that we would find the bad cytokines, which is all I knew about cytokines when we actually did this. And if you'd asked me what are the bad cytokines, I would have said TNF alpha. Isn't that like the only cytokine? And of course, there are thousands, it seems like. So we ordered a cytokine ELISA with a bunch of different stuff. I actually called the company, and I was like, I want the bad cytokine kit. And so that's how we started with this. So we started, you'll note there's no difference in TNF alpha, there's no difference in IL-6, there's no difference in IL-4, no difference in interferon gamma. Somebody suggested that we look at IL-10 because that is a quote unquote good cytokine, and that we would see that it is lower in our ApoC3 mice and that they would have lost a protective mechanism. And so to our surprise, we found that circulating IL-10 levels in our ApoC3 transgenic mice were 100-fold higher in our mice. And we've since measured IL-10 in a lot of different capacities, and it's really maintained. So we started to get excited because this was really opposite of what we would have expected. So where's the IL-10 coming from and what is it doing? What I was told is that a great source of IL-10 are Tregs, maybe dendritic cells. We actually did, my grad student, Kayla Rodia, did just an unbelievably detailed analysis of all the immune cell populations in most anatomical locations in our mice. And the only differences that we ever saw were in Tregs. And so here I'm showing you splenic Tregs being increased in our ApoC3 transgenic mice. These Tregs express CCR9, which is the intestinal homing marker. And then when we look in the lamina propria and the mesenteric lymph nodes, we also see an increase in the accumulation of Tregs. And so we asked whether the accumulation of intestinal Tregs and the increase in IL-10 would actually protect the intestine of these animals from inflammation, since Tregs and IL-10, if you plug into PubMed, out will come colitis. And so that's what we were asking about, whether they're actually active. And so we used the DSS colitis model, which is 5 days of DSS in the drinking water, followed by 5 days of regular water. DSS induces an injury-like colitis in the colon. It is a model of colitis. Human colitis is more T-cell dominated, which is why we've now been expanding these studies into the RAG1 model. But in this case, what we saw is that compared to the dashed lines, which were animals maintained just on drinking water and chow, that the wild type animals in the open circles lose almost 20% of their body weight before they start to recover. The ApoC3 transgenic mice, however, lose significantly less body weight and they recover more quickly when they're on DSS. And we used the qualitative assessment, macroscopic damage score, in a blinded capacity. We had our mice and they were observed every day for shivering, lethargy, diarrhea, bleeding, and a few other qualitative pieces. And I always tell this story because I think it's important for rigor and reproducibility, but when we had students, and at this time we had a lot of undergrads, they were shadowing and involved, and they came and said, we're really having a hard time keeping this blinded because it's really obvious which are the wild type mice and which are the transgenics once we were repeating this experiment, because the wild types always got so sick and the transgenics were just, you know, happy in their cages, hanging out, eating, running around. So it was really a robust phenotype that we were able to observe. We looked at the expression of tight junction genes in the small intestine and colon, and we see that the ApoC3 transgenic mice maintain tight junction expression. And we measured colon length, which is another marker of colitis severity, which is again maintained in the ApoC3 transgenic. So we concluded that the ApoC3 transgenic mice are protected from DSS-induced colitis. They also maintain their plasma IL-10 levels in response to the DSS. And when we looked, we see a slight accumulation of mesenteric lymph node Tregs, which we now are really interested in getting closer to where we think the effect is happening, which is in the lamina propria. So we think, you know, even though we see this accumulation in the mesenteric lymph node, that we're actually getting specific Tregs that are hanging out in the lamina propria, presumably Ldlr positive, and doing their work. So next, we asked whether this Treg associated protection, so you know, we have not definitively proven whether Tregs are actually the protective factor, nor have we proven that those Tregs are secreting the IL-10 that's protecting these mice. What we have found is that the accumulation of Tregs and IL-10 really correspond with the protection from DSS colitis. And so we asked whether that was due to ApoC3 itself or ApoC3's inhibition of triglyceride endocytosis. So though ApoC3 is a potent inhibitor of LDLR, it's not a complete inhibitor. And so we thought, would an LDLR knockout animal have a similar phenotype? And so that's what we did next, using wild type and LDLR knockout mice. We see that, again, the wild type mice lose weight in response to DSS, whereas the LDLR knockouts actually retain their body weight and come back to normal weight more quickly. The LDLR knockouts also have a decrease in that qualitative assessment, the macroscopic damage score. They are less sick. And they have an increase in both mesenteric lymph node Tregs and actually in the CD4 T cells in the mesenteric lymph node, which we're trying to sort out whether these are, which subset these total CD4s actually become. And so we were really, excited about this. We thought, wow, maybe raising plasma lipids or plasma C3 levels or inhibiting LDLR could actually increase this population of immune cells in a way that is protective. And so we asked whether there could be therapeutic treatment of mice with ApoC3 containing chylomicrons. And so what I'm showing you here are animals, wild type mice, who were simply given an IP injection of PBS, that's in the black box. And then in the gray boxes are mice that received an IP injection of purified ApoC3. This is lipid-free ApoC3. Now, lipid-free ApoC3 is just going to get rapidly cleared by the liver after an IP injection. It's not necessarily going to instantly find its way onto lipoproteins, and it won't increase plasma secretion of VLDL and chylomicrons. So we kind of use that as a control for whether ApoC3 could be doing something kind of non-lipoprotein related. And then we also injected ApoC3 containing triglyceride-rich lipoproteins. So we got chylomicrons from donor animals and to which we added ApoC3. And what you can see here is that the mice that received the lipoproteins have the least loss of body weight and move towards healing quicker than either of these counterparts. And so we're really excited about this preliminary data in particular because it tells us a little bit about whether this is cell intrinsic or more likely a cell extrinsic phenomenon on Tregs and IL-10. When we looked at the macroscopic damage, again, we see that the animals that received lipoproteins with ApoC3 are protected and that they maintain, that they have a higher level of FOXP3 and IL-10 gene expression, which we conclude comes from the Tregs, but we really need to prove by flow cytometry. And so we went in the literature to ask whether ApoC3 had ever been associated with inflammatory bowel disease before, and there are very few papers, but we found the following. We found a study that showed that a reduction in the ApoC3 gene is one of the largest changes in ileal gene expression in when you compare healthy and Crohn's disease pediatric patients. I'm a little unsure of what to make of this. There's not a ton of ApoC3 in the ileum, but it's interesting that it is reduced in patients with Crohn's disease. The gene expression of ApoC3 and members of its gene cluster, the ApoA1, A4 gene cluster, are reduced in the colon and ileum of adult Crohn's disease patients. And then in this paper, they measured protein levels, which were also reduced during a flare-up of Crohn's disease. Again, the directionality is similar to in our mouse model. And then in a broader way, if you knock out HNF4 specifically in intestinal epithelial cells, that will also cause spontaneous intestinal inflammation. HNF4 is one of the key regulators of the A1, A4, C3 gene cluster. And so this suggested to us that perhaps there was the premise for asking whether C3 is a protective factor in human colitis and whether total loss of ApoC3 in a mouse model, or to take it even a step further, using some of those pharmaceutical approaches might actually be deleterious. And so we used our ApoC3 knockout colony, which is also on a C57 Black 6 background, and we exposed those mice to the DSS model. You can see that ApoC3 knockout in wild type mice have a very similar loss of body weight with no significant differences between groups. Where we did see differences was in the macroscopic damage score. Again, this qualitative assessment, which we did blind. We found that the ApoC3 knockout mice consistently were sicker in their cages. More blood in the stool, more lethargy, although we didn't see that translate to an increase in diarrheal weight loss. We looked at colonic lamina propria Tregs, and we do find that there are very few of these Tregs in the gut of the ApoC3 knockout mice, which would jive with them being more susceptible to DSS colitis. And so at this point, where we're at is we're really looking at a suite of data that suggest, one, that CD4 T cells can endocytose lipids from plasma lipoproteins, and that, in fact, this arm of our hypothesis is a physiological event that occurs, and that it's possibly very distinct from what happens in response to free fatty acids. We showed that excess ApoC3 and the loss of Ldlr stimulates a physiological context where you have high IL-10 and intestinal Treg accumulation. And I put a hashtag here, or a star here. The mechanism is unknown. We're really trying to figure this out. Like I said, this is really the state of the lab. Are the Tregs homing to the gut? Do they represent a totally unique population that's hanging out in the lamina propria, responding to dietary antigen? Is there an increase in differentiation of the CD4 parent population? We don't really know. We're really actively figuring that out. We have some hints that it may have to do with the formation of X Tregs, and that the ApoC3 transgenic mice may have a maintenance of the FOXP3 locus in their Tregs, but that is really forthcoming. And our data at this point also showed that ApoC3 knockout mice, in ApoC3 knockout mice, there may be serious consequences to lowering plasma ApoC3, and that those consequences might be an increase in colitis severity. And so, you know, if you're a patient who has active Crohn's or colitis, you might want to think twice before taking one of the anti-ApoC3 drugs to lower your plasma triglycerides. And so, again, what does this mean physiologically? Excuse me. We found that ApoC3 overexpression may actually be good for the gut, and that it does this through an increase in tolerogenic Treg accumulation. We also then asked what we think is the logical question, which is, well, Tregs are good for the gut. They are protective against colitis. They calm inflammation, which is also predicated on those previous studies in cardiovascular disease. We would also want anti-inflammation in the heart in models of atherosclerosis. And so this led us to really ask, is it possible that ApoC3 is also good for the heart through this Treg mechanism, despite the fact that ApoC3 so significantly raises plasma lipids? And so we went about asking whether Tregs might also be found in the aorta. And, I'm hoping for the early career folks, what you can see is that our lab has really had, we've really evolved how we think about immunology and how we think about cells. We start now having data that looks more like, a real immunologist here. We've started really looking at these populations. And so here is just our ability to isolate Tregs, which are a small population, from the aorta. So here are Tregs in the spleen. Here are Tregs in the mesenteric lymph node. And you see we can isolate a really robust population of Tregs from the aorta. We take the arch and the descending limb for anybody who's interested. So there are Tregs. These are just wild type mice. This is not during a cardiovascular disease model. We asked whether these also express Ldlr, and we found that compared to the spleen, where something like 8% of the Tregs express Ldlr, over a third of the Tregs that we find in the aorta express Ldlr. And so again, what this tells me is that places where there are lipoproteins, there are immune cells that express the Ldlr in order to interact with those lipoproteins. This was our first CVD model. In this, we used wild type and ApoC3 transgenic mice. And these received no virus, and we compared that to wild type and transgenic mice who received the dominant positive PCSK9 AAV. And so what this model does is effectively knock out Ldlr because of the way that PCSK9 regulates Ldlr trafficking to and from the membrane. You end up with a liver. I'm not going to say liver specific because, new data has really shown that this model, that PCSK9 is in a lot of places, but definitely a liver enriched model for Ldlr knockout. And then we compared these groups. So here's body weight over time. What we did is give these mice the AAV and then feed them a high fat diet. And then what you see here on the left are circulating triglycerides. You can see the ApoC3 transgenic mice plus the dominant positive PCSK9 have ultra high triglycerides compared to transgenic or wild types. And that there really is a gradient of triglyceride response, with ApoC3 still having a significant impact on triglycerides. If you compare this to plasma cholesterol here on the right, you can see that the animals that received virus really have significantly elevated plasma lipids. And I've left the statistics off here, but they do exist. I should add them. So you can see the elevation in plasma cholesterol, which is what we really wanted in order to really push the cardiovascular disease model. Then the mice received 12 weeks of Western diet, and then we isolated the aortas. And so at the time that we did these studies, we again were really interested in macrophages and monocytes, kind of to go along with my TNF alpha view of the world. I'll tell you a little bit about the experiment we're doing now, even though I can't tell you, I don't have the data. It's the animals are being sacrificed this month, but first I'll start here. So in the animals that did not receive the dominant positive PCSK9, there's no difference in aortic macrophage number. But when you look at the ApoC3 transgenic mice who did receive Western diet and the dominant positive PCSK9, there's a significant reduction in the total amount of aortic macrophages. And then when we do the very basic M1, M2 subsetting of those macrophages, which I know does not actually, doesn't capture the real heterogeneity of the macrophages. But when we just gate based on M1 and M2, you can see that there's a huge reduction in M1 macrophages in the ApoC3 transgenic aorta, which to me is really exciting because these are inflammatory, putatively inflammatory macrophages. So there's no change in the number of M2s. And then when we look at the total oil red O staining, you can just ignore what's on the right here. That's our ApoC3 knockouts that we just kind of did for fun. But if you look here in the transgenics on the left, there's no atherosclerosis measured by oil red O in the animals that didn't receive the PCSK9, but in the wild types and the transgenic, there's no difference in the amount of atherosclerosis, which is actually really exciting to us because the ApoC3 transgenic mouse has all of these lipids, just tons of lipids, right? So as I said at the beginning, here I was kind of jealously holding on to our ApoC3 transgenic mice, like, oh, all of you cardiovascular people should be using this wonderful model. Well, there's a reason they're not using the model, even if we don't know exactly why. These animals barely get the wild type level of atherosclerosis. There would be a terrible model of athero, despite the high lipids. And so what we really take away is it possible that the previous work I showed you, the inhibition of Treg Ldlr, and then the subsequent change to lipid metabolism in those Tregs, is it also cardioprotective because Tregs are also important in atherosclerosis? And so what we're doing now is we're really getting down to brass tacks with those Tregs. We're asking whether wild type mice who've received different Tregs, Ldlr knockout or Ldlr complete Tregs, and then have the loss of Ldlr in the liver to raise their plasma lipids, whether we get an accumulation of those Tregs in the aorta and whether you get protection in that model. And so I hope the next time that I talk to you, I can really come down more conclusively about whether C3 is good both for the gut and the heart, but that's really what we're working on answering right now. I think what our data really clearly shows is that there's a real lack of understanding of how the physiological transport of lipids interacts with our immune system, and particularly triglycerides and cholesterol from chylomicrons and VLDL in Tregs. And so hopefully we'll have this work out, in paper soon. But with that, I will conclude, and I'll thank my lab members, including my previous lab members who've done this work, and our funders, and I'd be delighted to take questions.
Okay, Ali, it was a great presentation. Thank you so much. And also thanks for sharing some of the ongoing experiments, the unpublished data. This is great. So for those of you who have questions, please raise your hand. I know Ebru wants to leave, so I'll just give it to Ebru first. Ebru.
Ali, thank you so much. And beyond the fascination with the science, I'm really grateful you told us about the whole thinking process of how you evaluated the data and how one thing led to another, and that science is not a continuous linear path. So I think that's really important to remind ourselves. So thank you for that. And I was wondering, because I work in atherosclerosis, so the Tregs being really important for the regression in plaques. Did you consider removing the high-fat diet after PCSK9 and see how your phenotype is affected with?
Yeah, so that's on deck. Actually, the experiment, the design of the experiment that we have going right now is that we've used the Ldlr, the Ionis Ldlr ASO, and then done 12 weeks of Western diet. And then we're evaluating Treg populations then, with the goal of then extending it withdraw the ASO so that we can get into that regression time period. For the exact reasons that you're bringing up, which is, we want there to be athero. We know that most people, by the time we're talking about athero and statins and therapy, they're well into their plaque. And then how can we use the tolerogenic Tregs to reduce that? So we're like halfway through that model, Ebru. And I think it's really important to think about.
Okay, Katie?
Thanks, Ali. That was amazing as usual. I'm wondering, I may have missed it, but so you said that the Tregs actually endocytose the VLDL particle or whatever the particles are. What mechanism is that? Do you know? Is it full engulfment of the part? Like, do they digest? What's going on there?
Yeah, that's such a good question. So we actually don't know that they endocytose the whole particle. We know they endocytose the triglyceride, or at least, if I'm being really critical, the BODIPI label that's right on there. Yeah. So that is, you've really identified one of the sites that we think is really weak in this story. So we're using right now a lot of radio-labeled lipoproteins to really get at the relative contribution of radio-labeled triglyceride or cholesterol,
right,
to the cellular lipid pool.
Right.
And so, at the beginning, I kind of showed some pretty preliminary cell culture where we've done ex vivo and naive T cells and natural Tregs and induced Tregs. That's because we're really trying to actively answer your question. And does it matter if it's a Chylo or a VLDL or an HDL or an LDL? I think, I would say yes, it probably does.
Right, because like even just the size of these different particles, the way, we know from macrophages who are professional phagocytosers, right? They deal with all these particles so differently. And then I just think of a T cell, like what's it doing? Like what does it even have to do anyway? So that's very cool.
Yeah, we've also gotten some looks inside of these Tregs by EM, and we're really interested in the cytosolic lipid droplets. Because, most cells they're going to take up lipid and burn it or store it. And, I think a lot of the current ideas about immune cells and immunometabolism are kind of this, like there's a straight line, like a fatty acid, it comes in, it goes to the mitochondria, it does something, the mitochondria burns it, or it doesn't take it up, instead it uses glucose. But like there's like a lot that's happening inside of the cell beyond that. I think, again, whether the whole particle gets taken up, whether it needs to be, go to a lysosome, or all that stuff is a total black box that we're obviously really interested in understanding.
Very
too.
Cool.
But we're starting with dual radio-labeled lipoproteins, and just like you said, we're really looking at the literature for how those things were parsed out in macrophages to point the way.
Okay, Marit?
Well, thank you, Ali, for such a great presentation. Of course, I heard the story a couple of times, but I was wondering, have you also thought about a role of CD36?
I have, but right now I'm like ignoring it. I don't think that it is possible that this mechanism is LDLR specific. I think it's more likely that just like each of those lipid transporters are important in different ways elsewhere, they're probably really important in the immune system too. So we haven't done the studies in CD36 knockouts. I've been kind of flirting with the idea of using trying to get my hands on the floxed LPL animals to ask the same question. Could we just knock out LPL in the FOXP3 or even the CD4 cells? But I think it is very likely that there would be some kind of phenotype. we just started with LDLR since that's such an important ApoC3 target.
No, I mean, it makes total sense. And I just was only mostly asking the question because of the recent paper that came out where CD36 has a role in the expansion of the Tregs. And then just one other short question, because the ApoC3, you basically get these very large VLDL particles that are too large to enter the vessel wall. I thought that was the idea, right?
Yep.
Yeah.
So when we first did this athero study, I think, I was almost hoping, well before the our Treg stuff, I was kind of wanting or hoping, I guess, to see in person whether those particles would be so big that they would basically be, the endothelium would be like, blind to them. But I thought that can't possibly be the only, there have to be more things going on. And, I was like starting up my lab and I had gotten this advice, very good advice from David Huey, actually, in Cincinnati. And he said, when you're starting up your lab, find some great experiments and just redo them. Just old stuff that you think is dogma, redo it and really think deeply about it. And that was one of the, things was this athero study. And, it's good advice.
No, I mean, I think it's really a spectacular increase in IL-10 and also very nice story on the Tregs. Okay, thank you very much.
Thanks, Merit.
Okay, Esther?
Yeah, great story. I really enjoyed it. It's really fascinating that you get such an increase in Tregs. And do you know how they get there? You already told us that you were trying to sort that out, but are they being recruited there? Oh, sorry, these are my dogs. Are they really recruited into the intestine, or do they come from the intestine? Because the intestinal mucosa also, of course, has a lot of Tregs to kind of dampen the immune response. Do you have any clue?
Yeah, so we have a clue from negative data. So we had been using the a lineage tracing model where the Tregs are doubly labeled tomato GFP positive, and we've been using that in part to look at our X Tregs, and it's we haven't had great luck with the model. But one of the things that is either just not optimized in our hand or is true negative data is that we don't see a lot of homing from the adoptively transferred cells. And so when we look in spleen and see the CCR9 expression, I'm wondering now whether that's kind of almost like a secondary population of Tregs that are moving in after the first lamina propria events. And now I am collaborating with Tim Hand's lab here in Pittsburgh, and he's really an expert in these lamina propria resident Tregs. And we're trying to really find out whether, so at first we thought our Tregs really weren't a special population. It was just an accumulation of Tregs. And we looked at all of the markers, and I didn't show you guys this data, but we looked at CTLA4 and PD1 and Helios and all the stuff and didn't really see a huge difference. But when we did that from mesenteric lymph node Tregs, not lamina propria Tregs. And so when I, in my mind, put that together with the lack of homing that we're seeing in early experiments, it suggests to me that maybe the lamina propria Tregs are the population of interest, and whether they are expanding or proliferating, that's really something we're interested in.
Quite cool.
Yeah. Thanks.
Hey, next, Holger.
Hi, Ali. Yeah, that was wonderful. And actually, Esther already took my question kind of, because I was also thinking about the gut tropism and if the dendritic cells might be a little bit involved, because lamina propria dendritic cells express a lot of retinoic acid, and that induces this homing into the gut. And if that is kind of elevated, or the function of these DCs is maybe changed in
It's such a good question, and I really have no idea. We measured the CD103 positives in the mesenteric lymph node and in spleen and in whole tissue, whole small intestine, which isn't really ideal. And when we saw no differences, actually, we've also measured IL-10 positive dendritic cells in those locations. But what I've come to realize is that we're missing a whole site of action in the lamina propria. One of the ways that Tim Hand and I have been talking about recently, as we've begun a really nice collaboration, is the idea that triglyceride or even cholesterol sensing or metabolism by lamina propria, or let's just say gut immune cells, might be a way for those immune cells to know whether the mucosal lining is intact and normal. So I think there's a lot of, again, mucosal immunologists are the experts at this, but this idea that the antigens are coming through and there's all this back and forth antigen presentation and activation and all that stuff. But the small intestine in particular, it's a major function of the small intestine is making these chylomicrons and moving triglyceride through. So it seems logical to me that triglyceride may not only be a substrate for their metabolism, but is also a signal that the small intestine is healthy and doing the things it's supposed to do. And so in models where the immune cells either cannot access that triglyceride, like the Ldlr knockouts, or maybe CD36 knockouts, or these other knockouts where they can't access it to those cells, it seems like, oh, the small intestine is not functional. We aren't getting triglyceride. Or in our ApoC3 transgenic model, where there is actually pretty perturbed chylomicron secretion coupled with ApoC3 inhibition of Ldlr, go to the gut, go to the small intestine. That's another question that I get asked a lot. Colitis is a colon model. You're measuring small intestinal lamina propria, right? But it actually makes a lot of sense because that's where the triglyceride is coming through on chylomicrons. And so I'm really starting to ask, I think, even more basic question than a metabolism question. And it's just, hey, if the small intestine can move triglyceride, the small intestine is happy and healthy in that sense. If it can't, immune cells need to know that, and it can't, this would be another mechanism to tune them up.
And yeah, that's fascinating. And can I ask you once more if you already did some CNS2 profiling for the methylation status of these Tregs and the FOXP3 promoter region?
So, no, but that's very interesting.
The natural Tregs really have a demethylated CNS2 state.
Okay.
And the iTregs really will have a methylation pattern still there. Okay. And that's why you sometimes also see lower FOXP3 expression in these cells and more like a tendency to reduce IL-10, because the FOXP3 will be a little less stable in the iTregs, and they have a lower, or they are a little bit more fragile towards the environment. So I was wondering if you have really just the conversion of CD40 cells in the iTregs.
That's a great idea. That's a great idea. Thank you. Maybe I'll email you after this.
Sure. Yeah. Thank you.
Thanks.
Okay. Next, Mary.
Thank you. That was really wonderful. I love Tregs. But I, like Merritt, was thinking of some recent data I've heard from Ira Goldberg, where he shows that SRB1 takes up whole chylomicrons. Now this is endothelial cells, but do your Tregs express much SRB1?
I don't know off the top of my head because I don't know that we've looked at that specifically. We have some, I'm certain somebody has looked at it, but I don't know. I'm going to guess that they would. You know, this is what I think our data show is that any cell that uses lipids as fuel, which is every cell, has kind of a mechanism for taking them up and metabolizing them. And that if we haven't seen it in an immune cell, it's because we just haven't measured it. Because everything that we measure in our Tregs is essentially what I would expect in any other kind of cell. So I would expect that they do have SRB1, but
okay.
yeah,
I throw it out because he shows it very convincingly, and we've sent him knockout mice, and it really does block. And he does like an in vivo injection of your label Kylos. I think it's kind of cool that everybody's doing this, and it really does seem to block it. But anyway, you got some cool data, and keep going, girl.
Mary.
Okay, Ada?
Hi, yeah, I want to echo everybody saying this was a great presentation. My question is about kind of later responses because, you know, colitis is a huge risk factor for cancer. So I wonder, you know, if you looked at later times in the intestine, and whether if you only have the increase in C3, you know, in the later phases, also if that influences development of cancer.
Yeah, so that's a good question. We have not done any of those experiments. And my understanding of the DSS model is that if you want it to progress towards a cancer model, it's like multiple rounds of DSS in a row, which again would capitulate human colitis. My hypothesis would be that it would be like the opposite of the immunotherapy situation. So in cancer immunotherapy, you ramp up effector T cells. And so it's attack, right? Inflammation on high. And that's how you kill those cancer cells. The peripheral damage is then that you get attack of the heart, right? And that's what we see in the bad cardiac events in response to immunotherapy. I think the opposite would be true in our model. So you get Treg accumulation, high IL-10. This is all anti-inflammatory, which is excellent in the context of hyperinflammation. But in the context of cancer, I think it would be more, it would allow more cancer to thrive because you would have an inhibition of the effector T cells and whatever their role is in patrolling against colon cancer. And so we've thought about it, but just almost, you know, as a, you know, fun experiment, but not something that's like high on the list because I don't really understand it so well. But I think what I'm learning from immunology is that it's often a rheostat, but every time you turn it in one direction, you get the opposite direction now has the chance to flourish. So it's just so complicated.
Okay, great. So Ali, last question from me. You show a really nice pathway centering on IL-10, right? Did you check other anti-inflammatory cytokines or pro-resolving mediators like resolvents, specialized pro-resolving mediators?
We haven't looked at any of the resolvents. We will every now and again look at TGF-beta, which generally goes in the same direction as IL-10, but it's not as big of an effect. We are actually using right now an IL-10 knockout model to try to really prove that the IL-10 we're using a couple approaches, but we're trying to prove that IL-10 is the protective cytokine in our model, in the ApoC3 model. And then we're using other approaches to really prove that it's the Tregs. And then together it means that Treg IL-10 is the critical protective factor, but I'm sure it's not the only one. So that's where we are with that, but we haven't looked at the resolvents at all.
Right. Yeah. So Gabby would be the person, right?
Yeah, exactly. I was just going to say, I hope Gabby's not listening.
And last, like this Tregs are important in adipose inflammation. It would be interesting in metabolism, right? In obesity-induced adipose inflammation, what happens to these mice, right? In terms of a high-fat diet and then their systemic glucose metabolism. It would be interesting in the future for you if you're interested, right?
Yeah, no, I absolutely think that that's super neat because, in effect, our ApoC3 transgenic mice aren't sick. They're not ill, right? They're like in some ways, like the definition of like healthy fat, even though they don't actually get fat, but they've got these, really high lipids, but they don't really get any of the massive disease that we would think that they should get. And so I agree with you. I think that model would be very cool. And we did look at Tregs in adipose, but not after high fat diet, just in the basal state. We didn't see a difference, but that adipose tissue wouldn't have been inflamed. So.
Right. Okay, wonderful. Wow, that was a great discussion and an amazing talk again, Ali. Thank you so much. Thanks for all the great questions, and we'll see you next week. Bye, everyone.
Thanks, guys.